sexual reproduction in flowering plants. So first of all, sexual reproduction means with the help of gametes. Whatever the reproduction is going to take place, that is the male and female gamete, it's called as a sexual reproduction. Okay, reproduction means what? It is the generation or it is the formation of what? New offspring. That is called as what? Reproduction. Formation of offspring. Whereas flowering, okay, flowering means what? Flowering means generally flowers, whatever is grown. Okay, so that process where the flower grows is called as what? Flowering. So mostly all the flowering plants which are there, they are generally termed as what? Angiosperms. Suppose if they are non-flowering, then they will be termed as what? Gymnosperms. I'll just write it over here. Fine. So these flowering plants means only they are going to show what kind of reproduction? The sexual reproduction. And what are the sites of sexual reproduction for the flowering plants? It's the flower. So flowers are what? The sites of sexual reproduction. Now, obviously, whenever there is a fertilization taking place or when the structures are formed, then there will be certain what changes. So what type of changes can we generally see? Okay, so basically, the changes that we can see is, I'll just write it over here. Yes. So they are called as pre-fertilization events. So in this, there will be what? Changes in certain structures as well as what? The hormonal changes. So whenever there is a several, what do you say, hormonal or structural changes, then that results into what? Differentiation and development. Of what? Because we are learning about the flowering plants, so it will lead to the differentiation and development of the floral primordium. Okay, means the entire flowering plant which is there, that one. Okay, so let's see one by one the pre-fertilization structures as well as the events which going to take place. Okay, now usually the floral buds which are there or the things which produce the flowers, they are called as what? Inflorescence. Inflorescence. Okay, so this inflorescence, they will bear what? The floral buds. And then it will form flower. Fine. So basically, we'll be seeing in detail about the structure of the flower. So first, let's see what is the actual structure of what? The flower. Structure of flower. Now, basic things we already know that how does a flower actually look like. Okay, like it'll be having petals, then we'll be having the stamens, carpel, all those stuff. So one by one, we'll be seeing all the structures. So if you take a typical flower, that flower will be having what? Basically two parts. Okay, so basic two parts, that is the androecium and the gynoecium. So androecium is the male reproductive part, whereas the gynoecium is the female reproductive part. Okay, so first let's see about the male reproductive part, which is nothing but the androecium. Okay, so androecium. So first of all, it's the male reproductive part, first thing. Second is what? It consists of a wall of what? Stamens. See, when it's a male gamete, then it's stamen. When it's a female gamete, it's called as carpel. Okay, these are very important terms. So this particular androecium which is there, it is going to have what? A wall of the stamens. Okay, so that particular stamens number as well as length which is there, it will vary according to what type of what do you say, flower species they are. Okay, so whatever the stamen is there, they will vary in what? Number and length. Now this all depends upon the species, whatever species they are into. So this stamen is going to have what? Basically two parts. Okay, so like if you see a flower, in that this part which is there, okay, this part is only the entire stamen, or what you can say is the androecium, the male reproductive part. Fine. So whatever the stalk like thing which you can see over here, this is called as the stalk, is the common name, but it's also called as filament. Okay, and the upper one which you have, these two things which you can see over here, right, that is called as what? Anther. Fine. So this is what a general basic structure of what the male gamete androecium. So coming towards the two parts of a stamen, right? So the first part of a stamen is called as what? Filament, or a stalk, basically. So what is this filament and or a stalk? So as per the diagram, you can see it is nothing but what? A long and slender stalk. Okay, so whatever the proximity of or the proximal end of that particular filament is there, that will be attached to what? The thalamus. Okay, so the green color part, bulgy part which you see in a flower, right? So that is only called as what? Thalamus. So here the thalamus will be present like this. Okay, and here only that, like this it will be there. If you see, okay, so filaments are nothing but a long and slender stalk, okay, whose end is being attached to whom? The thalamus, proximal end. Proximal end attached to thalamus. Or either you can say the petal of the flower. Okay, so from here only the petals also will be starting. Okay, so this is the first part of a stamen. Now the second part is called as what? Anther. So in the diagram, already you can see here, if you see this diagram, here there are what? Two, like this thing. So this is called as what? Bilobed structure, because two parts are there. So hence they are called as what? Bilobed structure. Bilobed. Okay. So each of this lobe is going to have what? Two thecae. So two thecae means because it's bilobed, right? So generally it is termed as what? Dithecus. Dithecus. Okay, so this is basically a longitudinal groove which is generally going to run in what do you say, between this structure, and which is going to separate these two thecas. These two which are there, right? So this is called as theca. So because there are two thecas, hence it's called as what? Bilobe structure, but with two thecas means dithecus. That is the term which is used over here. Dithecus. Okay, so this particular anther part which is there, that is in detail actually. Okay, so let's see the anther part in the detail. Okay, so already we know that bilobe structure, a dithecous structure it is. But one more thing is there, that it is also what? Tetragonal. Now tetra means four. So it is basically what? Tetragonal structure. Means it is consisting of four things. Okay, that is called as what? Microsporangia. Now, first of all, let me tell you that whatever the male gamete which is present in the plant, right? So that is nothing but the anther, right? So pollen grains which are there, they are generally referred to as a male gamete. So this pollen grains only when it comes into the flower, then only whatever the further fertilization, reproduction events which are there, it will take place. Okay, so for this purpose, for the pollen grains, okay, this particular microsporangia which is there, it is very important. Okay, so in each of this lobe over here, okay, in each of this lobe, that is two lobes are there, right? So if I do it like this, zoom it. So here, just consider this as four parts like this. Okay, right? So this is only altogether called as what? The four microsporangia. So they will be located where? In the corners. In the corners of the, like each of the lobe, you will find this what? Microsporangia. So this particular microsporangia only develop into what? Pollen sacs. Pollen sacs. Right? So they are going to extend, okay, longitudinally, means whatever the lengthwise, throughout the length of that particular anther, they will be what? Extending. And this particular pollen sacs is going to have what? Pollen grains, which is the main male gamete. Okay, so whatever the sporangia is there, that is going to develop into the pollen sacs, and these are the pollen sacs which will contain what? Your pollen grains. Okay, so this is what in general, whatever the anther is there. Now moving on towards the detailed part of this microsporangia. Fine. So if you see a microsporangium, okay, like whenever you're drawing that particular thing, it'll be somewhat like this. Fine. So here, because there are four layers, as I told you, tetragonal it is. So like this, it will appear. Okay, and then obviously it'll be having certain layers like this. Okay, 2-3 layers it will be having. Right. Okay, like this 2-3 layers it will be having. So like if you see this part, this is called as the connective. Okay, the first part is the epidermis, the first layer. Okay, then you have the middle part, which is called as what? Endothecium. Then this four circular things which you can see over here, they are nothing but the sporangious tissue. Sporanginous tissue. Then the outer layer of this particular sporanginous tissue is termed as tapetum, a very important point. Okay, tapetum. And then whatever over here you have is called as the middle layers. Middle layers. So epi means outer, endo means inner, and middle layer is middle. Okay, so this is how it'll appear generally whenever you take what? A transverse section of a sporangia. So microsporangia, this is how it will like appear. Okay, so now let's go to the structure of a microsporangium. So in this diagram only you can make clear, like make it out like this particular sporangium is near what? Nearly somewhat like circular in shape. Okay, because you can see a lot of circles are there. So it's generally what? Circular in shape. Okay, so that is shape in the sense the outline, whatever which is there, it will be circular. Okay, so the first point is what? Circular outline. Second points I told you what things that particular microsporangium consists of, right? So generally this particular thing is surrounded by what? Four wall layers, okay, which are nothing but the epidermis, endothecium, middle layer, and one more which is important I told you, that is the tapetum. Okay, so tapetum is generally the innermost layer of a microsporangia. Okay, so whatever the three outer layers which are there, that is the epidermis, endothecium, and middle layer, what they are going to do is they are generally going to give you protection. They are generally going to give what? The production. Plus, they will help in what? Dehiscence. Dehiscence of anther. Now, what is dehiscence of anther? Now, see, that particular pollen sac or the microsporangia contains what? The male gamete, which is pollen grains. So, pollen grains has to be released or squeezed, right? So, that is only called as what? Dehiscence of the anther, means the stimulation it will give. Okay, so for what purpose will it give the dehiscence of the anther? So that it can release the pollen grains. Release the pollens. Then what about the tapetum? Now, tapetum is generally for what? Nourishment. To whom? To obviously the developing pollen grains. Correct? So, this particular tapetum cells which are there, they will have what? Cytoplasm plus one nucleus. Now, sometimes one or sometimes more than one. Okay, one or more nucleus, you can say. Okay, so what happens is whenever this particular anther is in young stage, okay, when it's in a very small stage, at that time, whatever the sporangium is there, it is going to have this circles are there, right? Which are called as what? Sporanginous tissue. So whenever the anther is in younger stage, it will be having what? A sporanginous tissue at the center. Okay, so third point is what? When it's younger stage, anther, it has sporanginous tissue at center. Okay, so the sporanginous tissue is nothing but it is going to have what? Diploid sporogenous cells. Sporogenous cells. Right? So now obviously for the pollen grains to be produced, there has to occur some kind of processes, right? So that particular process is called as what? Microsporogenesis. Sporogenesis. Genesis means formation. Microspores, you already know, it's the pollen grains. Okay. So what is this microsporogenesis? Formation of the microspores are called as what? Microsporogenesis. So what happens is when the anther develops, okay, each of that cell, which cell? The sporanginous cells which are there. Okay. Each of the sporogenous cell. Now, these sporogenous cells are also called as what? Microspore mother cell or pollen mother cell. So, this mother cell is going to give rise to what? Your ultimate pollen grains. Okay, so what happens is whenever from the young stage when the anther goes on maturing or developing, whatever the sporogenous cells which are there, they will undergo what? Meiotic division. So when it will undergo the meiotic divisions, then it is going to form the microspore tetrads. That is the 4 cells I told you, right? So they are the microspore tetrads. So whatever the formation of microspores are there, they are from whom? This PMC, which is nothing but the pollen mother cell. So whenever there is a formation of microspore from the pollen mother cell through the process of the meiosis, it is termed as microsporogenesis. Okay, so once this microsporogenesis has occurred, next what is going to happen? The anther is maturing. So when it matures, it is going to dehydrate, and then whatever microspores are there, they will develop into pollen grains. Right? So each sporangium, if you take, they will be having thousands of pollen grains like this. Okay, so whenever there is a bust of that anther, this pollen grains, it will be released and it will be going into each of the flower as per whatever ways it is. Okay, so this is all about what? Microsporogenesis. Okay, so now let's see the pollen grains and what parts does a pollen grain actually consist of. So, pollen grains, also known as the male gametophyte. So generally, you may have seen the pollen grains. How do they appear actually? They're generally what? Spherical in shape. Usually, their diameter will be 25 to 50 mm in diameter. That much small they will be. And they will also be surrounded by what? Cytoplasm. And this cytoplasm is surrounded by the plasma membrane. Same as the typical cell, whatever parts are there, here also it will be present. Okay, so this particular pollen grain is what? Two-layered wall. It has two layered walls. So what are the two layers called as? Exine and intine. In the name itself, you can see exine means what? Outer, intine means inner. Okay, so the exine which is there, they are what? The outer layer, and they are also a hard outer layer. Okay, it will be very hard. So, the reason why they are hard is because they are made up of a substance which is called as what? Sporopollenin. Now, this particular sporopollenin which is there, it is very resistant to what? Organic materials. Okay, so hence it is able to withstand whatever the higher temperatures outside which is there, or strong acids, alkalis, whatever is there, harsh chemicals which are there, right? So it can withstand all those stuffs. Okay, so this particular sporopollenin is there. It cannot be degraded by any type of enzyme. Okay, so hence the exine is called as what? Hard outer layer. Okay, so this exine also has what? Small apertures. They have what? Apertures means hole-like thing, which is called as what? Germ pores. Germ pores. So in this germ pores only, the sporopollen is absent. Thus, everywhere in the pollen grain, you will find this particular sporopollenin. Okay, so if you see, even you will find the fossils of the pollen grains. Okay, so how do we get the fossils of the pollen grains? Is due to this hard material which is present in them, which is the sporopollenin. Fine. So this is the first layer. Then comes the second layer, which is called as what? Intine. So intine means, as I told you, inner wall, right? So this particular inner wall, comparatively to the exine, it will be what? Very thin, and they will be having a continuous layer. And this continuous layer is made up of two things, which is cellulose and pectin. Okay, so here important points are in exine, sporopollenin is present, because of which it becomes, what do you say, resistant to many factors like temperature, alkali, acids, and so and so things. Whereas in the intine, there are two things present which are called as cellulose and pectin, which is going to make a continuous layer inside that particular intine. So these are the two layered walls of a pollen grains. So what happens is, whenever a pollen grain matures, okay, there will be two cells present in that particular matured pollen grains. Okay, so when this pollen grains matured pollen grain, it will be having what? Two cells. Okay, so the first one is called as the vegetative cell, and the second one is called as the generative cell. Right? So vegetative cell means they will be bigger in size. And this will be what? Smaller in size. Okay, so because the vegetative cells are bigger, lot of food can be accommodated into it. So hence it has what? Abundant food reserve. Means it can store more food. Okay, and plus it is what? Large and irregularly shaped nucleus it will be having. Then generative cells which are there, they are smaller. So because they are smaller, they have the capacity to float. Okay, where? In the cytoplasm of the vegetative cell. So here the shape over here will be the spindle shape. Okay, and it will also be having what? Cytoplasm plus the nucleus. Okay, so generally if you take a particular cell like this, okay, just roughly I'm drawing over here. So here inside like this, if you take, okay, so consider this as the nucleus of the like vegetative cell because it is a larger cell over here. If you can see, this is a smaller cell. So consider this as a generative cell. And this thing is what? The nucleus of the generative cell. Okay. So if you see, like out of 100 percentage of angiosperms, 60 percentage of angiosperms, they will shed their pollen grains at this stage. That is when they are in this stage with the vegetative cell and generative cell stage, they will shed their pollen grains. Okay, so what happens is whatever this generative cell is there, they are going to divide mitotically. Okay, so what happens is whatever the generative cell which are there, that is going to divide what? Mitotically. So mitotically when it divides, what it will give? Two male gametes. So whenever there is a shedding of the pollen grains, they will be shedded at three-celled stage. So whatever the shedded pollen grains are there, where they will land? They are going to land on stigma. So how long is it going to be alive? That will depend upon the temperatures, the humidity, or whatever the external factors are there. On that basis, it will depend. Sometimes the pollen grains may be alive just for maybe half an hour. Okay, sometimes more than that. So everything depends upon what? The temperature and the conditions which it is going to get. Okay, so this is all about what? The male gamete, pollen grains, okay, and the process which is there, how it is formed and what happens next.